Hello, everybody. This is Chelsea Miller, and this is my presentation on my microcosm that I built in Key West for General Ecology EVSP 416 for Professor Jesse Thornburg. All right, let's get this started. So, a basic microcosm overview. Microcosms are a unique way to study ecological interactions from various species, such as predation, competition, and mutualism, in a controlled environment. They can reveal how community structure and species diversity respond to disturbances or change over time. Microcosms can provide insight into processes like decomposition, sedimentation, and gas exchange under controlled conditions. And researchers can observe how nutrients like nitrogen, phosphorus, carbon, and others are cycled within ecosystems and how changes in nutrient availability affect ecosystem function. Long-term experiments in microcosms can help observe evolutionary responses to selective pressures and adaptations. Ecology and microcosms go hand in hand and can show us many different aspects of our world. They can serve as experimental setups for testing ecological theories and hypotheses under controlled conditions. They act as proxies to understand the functioning of larger, more complex ecosystems without needing to manipulate entire natural environments. Microcosms can better reflect the ecological effects of toxic substances like pesticides in a smaller, controlled environment. They help assess the ecological effects of human activities such as agricultural runoff, industrial pollution, or habitat fragmentation. They are also used to study the ecological effects of introducing or removing species, such as invasive species or key predators in smaller ecosystems. There's a picture attached on the left-hand side of the slide that shows the origin, development, and application of the microcosm. The left side of the picture shows the origin and development, while the right side shows the application. The left-hand side shows different points in history in 1982, 1986, and 1858. On the right-hand side, going down, the first picture shows the migration and degradation of chemical substances in the environment. B shows the ecotoxicology effects for chemical substances. C, the ecological risk assessment. D, bioremediation and ecological restoration assessment. E, energy transfer and bioaccumulation in the food chain. And finally, F, the interaction between organisms and the environment. So let's get into my individual microcosm. When I retrieved my sample, I did not have high hopes for a significant change. The vegetation in the bottle was all one type, and there were no visible arthropods. I believed that due to the lack of variety in the microcosm, that I would not see much change, if any. I took my sample from the Nature Preserve of Key West, with permission, of course. There was a small bridge that went out into the wetland about 10 meters where I retrieved my sample. This is a unique wetland area due to its proximity to the Atlantic Ocean, which can be seen in the picture on the right. The only thing separating the two habitats is a small strip of beach. I retrieved 1/2 cups of sediment and filled the jar to the 3/4 point with water. I then grabbed 5 plants floating on top of the water and a funny looking seed for variety. This will become very important later. On this slide are pictures that I took on the day of the sample retrieval at the sample site. The first picture on the left is a beautiful yellow-crowned night heron that was watching me take the samples. The second picture on the upper right is me collecting the sample in my awesome mushroom shirt. And the third picture on the bottom right is the view when looking out at the wetland if you were to stand up at the spot that I was taking the sample from. After sealing the microcosm, it was placed on my westward-facing balcony for 7 weeks. I live in Key West, Florida, so the temperature is very tropical and sunny, especially this time of year when are out of the hurricane season. The microcosm got direct sunlight for about 5 hours a day, and the rest of the day it had indirect sunlight. Over the course of the project, the average temperature lowered about 10 degrees Fahrenheit as our winter weather rolled in, but there were no days of extreme changes or variation. As you can see, before shutting the microcosm, I took a picture of the plants that I believe to be smooth frog bits, Limnobium laevigatum, and one floating seed of some sort that has a white circle with the green ring of algae around it. The turbidity of the water is relatively high, but not completely dark. Within a few weeks, I could see a white growth developing on top of the plants, and it was rapidly taking over the top layer. Visibility was also decreasing, and the water was becoming darker and harder to see through. There was a suspicious pink ring growing just above the sediment. It is hard to see in the picture here, but at first I had no idea what to make of it. I did not know what was to come. By week 6, this pink film had enveloped the entirety of the inside of the jar, and I could not see anything in the water. It was all a mystery of what was happening in there. The sediment on the bottom was turning a bright green as well. After researching, I found that the pink residue could be some form of bacteria or algae. Since I could not see anything that was happening inside of the jar, and we were at the end of the experiment, I decided to open it. When I did, it was pretty well pressurized, and I had a hard time opening it. I also might have gotten some type of mold poisoning. Only time will tell. It was pretty rancid, and all of the organic matter was consumed by mold and algae. I believe this was due to that tiny floating seed that I added, which had some type of mold already growing on it. The plants were no longer alive, and it seems that a multicolored growth had developed all of the organic matter. This experiment went a complete different way than what I had hypothesized it was going to go, and thus my hypothesis was incorrect. The addition of one tiny element completely changed the entire ecosystem. The dominance of microbial life over plant matter may have implications for understanding nutrient overloading and eutrophication in larger wetland systems. The conclusions of this experiment include species interaction, nutrient cycling, and anthropogenic simulations. For species interactions, the introduction of a new species significantly influenced the community. The rapid growth of mold and algae highlights how certain organisms can dominate in a closed system. For nutrient cycling, the green sediment and pink bacterial or algal film suggest significant nutrient redistribution, possibly driven by decomposition and microbial growth. For anthropogenic simulation, the introduction of the seed mirrors real-world scenarios where external inputs, like invasive species or pollutants, can significantly disrupt ecosystems. This was a fun experiment, and I was pleasantly surprised to see what happened to my ecosystem. I think further research of a similar wetland site would be nice to see, one that I can get arthropods from and really make it a thriving microcosm. The next time I do this, I probably won't take an item that looks like it isn't part of the naturally occurring habitat. Who knows where that seed came from? Thank you, everybody, for listening to my presentation. Just for fun, I added some pictures of a second microcosm that I had going at the end of my presentation. This one did not get demolished by eutrophication. Thank you very much. Have a good day. These are my references. I didn't know how to continue the PowerPoint, so uh, if you want to read them, you can. On to the bonus microcosm. This is my second microcosm. It was taken at a botanical gardens here in Key West. I actually volunteer there on the weekends sometimes, so that was pretty easy to get. So this was actually retrieved in a body of fresh water, and a fun fact about it is that underneath the water, it actually started off as a burial site for the military's like fuel storage tanks that they were no longer using. So they cleaned that up somehow, and now it's actually a nature preserve, and it's a pretty beautiful place. But all right, onward. This slide is just going to show you pictures of the location that I got the sample. The upper left is a zoomed in photo of where the sample came from. The bottom left is a zoomed out photo of where it came from. And on the right is the local turtle population. They're a bunch of red-eared sliders that just come right up to you, and you can feed them little flowers, and they're sweet little guys. So for this microcosm, I was able to get more of a variety of vegetation. I got some rooty plants along the bottom, along with some algae, and actually like rooted in plants that were kind of stuck to the bottom as well. I got them from a couple different spots within the same about 20 feet or so. I had expected this microcosm to actually have a lot of variation and change throughout the next few weeks, but I was pleasantly surprised. So, like I said, I was surprised. This microcosm did not really change at all. There was some algae growth, which you can kind of see on the right picture there on the far right side of the jar, and a little bit on the left picture too. This is the last week, so today I actually took this picture, and uh, there wasn't a whole lot of change. The plants look like they're still alive in there. It didn't get decimated by bacteria and fungus or algae. So I'm actually going to keep this one. I did not open it to kind of see if it continues to do anything. So also, I don't feel like this is a biohazard as well. So this little guy's going to stay on my porch. But if you stuck around long enough to see all of this, thank you for coming by, and I appreciate everything you guys do and science. Yes.